Welcome to the Brookings AP Biology video PowerPoint. This is a PowerPoint modified from one taken from Kim Foglia's explorebiology.com, so I have to send a thank you out to her. What we're going to talk about today is macromolecules. Molecules, the four major molecules found in living things. And you learned about them in bio: carbohydrates, lipids, proteins, nucleic acids. So we're not going to spend a lot of time going back over the things you should already know. If you don't remember doing this in bio, go back and look at the bio link and review a little bit. But we are going to go on and add some new information to that, what you already know. The first idea we're going to talk about and refresh your brain about is the idea of making polymers. Polymers are large molecules made by joining smaller little pieces, similar subunits together to make a bigger molecule. And we join them together by making covalent bonds holding those subunits together. The little pieces are called monomers. We learned that as a vocab word in bio. And the big molecule made from those monomer subunits is called a polymer. Lots of biomolecules are made, those pieces put together, using a condensation reaction called dehydration synthesis. We learned about this in bio, and if you go back and review your bio stuff, if you want to see it again or learn a little bit, refresh your brain. This is a reaction where you're taking a hydrogen from one molecule and an OH from another molecule and removing them. We're pulling out those atoms to make a water molecule, and what's left behind joins together to make a covalent bond. One molecule provides the H, one provides the OH, and you're removing, dehydrating, you're removing water to synthesize, build something. This requires energy and enzymes. It's a chemical reaction. And we're going to watch a little animation about how this works. If you look at molecules, and this could be any kind of a molecule in your body. Most organic molecules have oxygens and hydrogens in them, so we're not going to worry about what the rest of this molecule is looking like. As long as you have O's and H's, these two little pieces, monomer pieces, can join together using dehydration synthesis. We're going to pull out a water molecule and join and make a bond between the subunits. Remove water, dehydrate, synthesis, we're putting something together. Removing a water molecule to join smaller monomer pieces. And depending on what those monomers are, we can make different kinds of molecules. The reverse of that is hydrolysis. We use it to take a bigger molecule and break it apart. Remember, if I made a bond by pulling out a water molecule, I could break that bond again by adding the water back in to where the bond was. So I add the H and the OH back in, and I can break apart a bigger polymer into its smaller subunits. We use this in digestion when we're breaking down food. And if we go back to our little animation again, we're going to do hydrolysis this time, taking a bigger molecule, add the water back, and break the pieces apart, remove the subunits. That's hydrolysis. Hydro, water, lysis means to split or break something. And so we're going to do hydrolysis. Notice those, excuse me, those two reactions are kind of the opposite of each other, and they work together in your body to build up molecules and break apart molecules. The first group we're going to look at in this slideshow is carbohydrates, and we talked about all of these groups in biology. Carbs include things like sugar in your food, starches, and we're going to talk a little bit about what they look like, how they're made. The smallest little kind of carbohydrate is a monosaccharide. Mono means one. Saccharose comes from the Latin word meaning sweet, one sugar. It's a simple sugar molecule. And these kind of carbs have the same combination or the same ratios of carbon to hydrogen to oxygen. They can have different numbers of carbons, but they have that 1 carbon to 2 hydrogens to 1 oxygen. So I could have a 3 carbon sugar like glyceraldehyde, 3, 6, 3, 3 carbons, 6 hydrogens, 3 oxygens. I can have 5 carbon sugars, we call them pentoses. We can have 4 carbon sugars, 6 carbon sugars. Glucose is a good example. If you look at these 6 carbon sugars, notice they all have the same formula, C6H12O6, twice as many hydrogens as carbons and oxygens. These are structural isomers. We talked about that in our last chapter when we looked at carbon and making isomers. So 3 to 7 carbons usually in a sugar, and oftentimes the ends of the name ends in the letters OSE. Look at the names glucose, fructose, galactose, ribose. Oftentimes sugar molecules, a clue that it's a sugar molecule, it has "ose" at the end of its name. When we talk about molecules and look at how they're put together and how they make bonds and interact with each other, it's important to know which carbon we're talking about. And so, for example, glucose, I can number the carbons, carbon 1, carbon 2, and when we start numbering in a glucose, we always start with the carbon at the end that has that carbonyl group on it. So this is carbon number 1, carbon 2, carbon 3. Even when sugars form rings, and we're going to talk about that, instead of straight chain structures, we can still name and label those carbons. Here's our carbon number 1, carbon number 2, 3, 4, 5, 6. Notice that first carbon came from this carbon here that has that carbonyl group on. If we look at a glucose molecule, remember we talked about asymmetric or chiral centers in atoms or in molecules that have carbon as part of their structure. If you look at this carbon here, notice it is hooked to 4 different things. That makes it an asymmetric carbon. It has an OH, it has an H, it has this group here, and it has this group here. 4 different things. So that means if it's a chiral, an asymmetric carbon, it can have stereoisomers. There's 2 forms of glucose. And remember we talked about isomers, the D form and the L form has to do with how they rotate light. Dextrorotatory or levorotatory. But what we're going to look at is those 2 isomers, remember we talked about stereoisomers, are designated either D-glucose or L-glucose. The interesting thing is most naturally occurring sugars are the D form, the D isomer of those 2 stereoisomers. D and L, remember, are mirror images of each other. We made some of those using molecular models. Pentoses and hexoses, ones that have 5 carbons or 6 carbons, can cyclize when they get into water. That means they change from that linear form into a ring form. And we're going to watch a little animation about how that works. If we take glucose and we put it in water, which basically means inside of a cell, remember cells are mostly water. Here comes my glucose molecule. Notice the ring form, when it gets into water, can change shape and cyclize. It makes a ring. And depending on how that bond forms, the OH can stick down or it can stick up. And both of those forms are produced when you cyclize and put sugar into water. So if the hydroxide group ends up hanging down below the plane of the ring, we call it alpha glucose, alpha D. Remember, D is the stereoisomer form. Or if it ends up sticking up above the plane of the ring, we call it beta glucose. Both of those forms are found in living things. And remember, we can number the carbons, starting with that carbonyl group, and if you look at how those carbons end up when we get to our ring, you can see where those numbers end up in the ring. We can label the carbons, and that's going to be important when we talk about bonding in just a second. What do we use carbs for? They're our energy supply. If your body is a car, glucose is the gasoline that you put in. Cells burn glucose in their mitochondria, and they store the energy that's released as ATP. We learned about that in bio. The other thing when we talk about carbs is not just simple sugars, but we also can join sugar molecules together, those monomers, monosaccharides, and we can use dehydration synthesis again to join pieces together to make bigger sugar molecules. For example, disaccharides, two sugars, two sweets, are made by joining two sugar molecules together to make a bigger sugar. The bond that forms between sugar molecules is called a glycosidic linkage. An example, I can take glucose and fructose, two different sugars, and join them together to make a two sugar molecule, a disaccharide called sucrose. It's one of the most common sugars. It's table sugar. You sprinkle it on your cornflakes, right? It's the most common disaccharide, and it's made using that dehydration synthesis reaction. I can take sugars and join them together to make even bigger molecules. So different combinations of subunits, different kinds of sugars joined together make different kinds of disaccharides. For example, glucose and fructose, we talked about that in the last slide, makes sucrose, table sugar. Two glucose sugar molecules together, added together, make maltose. Glucose and galactose make lactose, milk sugar. Some of you may be lactose intolerant. That's the inability to break down that sugar that's in milk when you eat milk products. If I take lots of sugar molecules and hook them together, I get what we call a polysaccharide. Poly means many, many sugars. An example that we learned about in biology was starch. And we talked about structurally how is starch and glycogen and cellulose, the polysaccharides we learned about in bio, different, is how they are attached together. We're still using glucose to make them, but how they hook together is what gives us our different shapes and what gives us our different functions. So if you look at starch, starch is a polymer using the alpha glucose, the one where the OH group hangs below the plane of the ring. So if you take alpha glucoses and you hook them together using dehydration synthesis, remember, pull out the water, make a bond, you make a many sugar molecule that is very, lots of sugars hooked together. And it's called a polysaccharide, many sugars. This is what plants use to store energy. And we have the enzymes to also break these bonds apart to use the energy that's in starch. When we eat things like pasta, bread, all of those things have starch in them. Potatoes, we can digest those bonds. Notice we also have some branching bonds that come off this number 6 carbon. So two forms of starch. One is very branched and one is not so much. The non-branching form of starch or less branching form of starch is called amylose. And the more branched form of starch, where we're making not only 1,4 bonds, but 1,6 bonds to make that branch up, is called amylopectin. Both of those are kinds of starch molecules made by plants. The other kind of polysaccharide that animals make, like you, is glycogen. Also notice we're making 1,4 glycosidic bonds like starch. It's more branched, it's more branchy, and more than the amylopectin. And this is the way that animals store their sugar for later. Animals store their sugar in their muscles, in their liver, and we join glucose molecules together to make glycogen. The third kind of polysaccharide you learn about in bio is cellulose. It's a major building block to make cell walls. when you crack a piece of celery and you see that stringy stuff along the stalk, that is cellulose. It's like a building, those I-beam metal beams that they put in a building before they add the bricks. It makes the building sturdy. It holds it up, and that's what cellulose does in plants. It's one of the most abundant compounds on Earth. Think about how many plants there are, and cellulose goes together a little different way than glycogen does. Notice now we're going to use the beta form of glucose to make cellulose. So beta, because the OH sticks up, it goes together in a little different way. And notice every other bond, upside down, up and down, up and down. It takes different enzymes to make that happen, and it takes different enzymes to make that break apart. The enzymes that digest starch by breaking those alpha linkage in starch don't work to break the beta linkages that are in cellulose. Cellulose, when we eat plants with cellulose in it, basically it passes through, we can't digest it, we talk about it as fiber in our diet. There are bacteria that do have the enzymes that break down cellulose, and they live in the digestive tract of animals to help them break down the plant material that they eat. Think about termites that eat wood. The termite itself does not have the enzymes to break down cellulose, but the bacteria that symbiotically live in its digestive tract work to help the break down the cellulose and allow the animal to absorb the molecules from them. The last kind of polysaccharide that we did not learn about in bio is chitin. Notice how it's spelled. It looks like chitin, but it's pronounced chitin. It's a structural building block polysaccharide like cellulose, made from beta glucose subunits attached together. What's different is, notice our ring also has this nitrogen group attached to the sugar. And this is an example of a polysaccharide, many glucoses hooked together, but the glucose has been modified a little bit with those nitrogen groups. Organisms that make chitin are things like exoskeletons in arthropods. They have chitin to help make them sturdy. They're found in fungi, put chitin in their cell walls. If you have ever had an operation where you use those dissolvable stitches, sometimes those are made out of chitin. It's a very strong and sturdy material. That is the end of our slideshow. We're going to talk about lipids in our next video, so stay tuned.